Antimicrobial resistance is a global health problem and we're all at risk. What we really need is much better antibacterial treatments, we need better vaccines as well as much faster diagnostics. That's why at the Novo Nordisk Foundation we are very proud to support CARB-X, which is accelerating innovation across these areas.
We have supported and advanced more than 100 high-value projects in this space. All of them target the pathogens of highest unmet need on both the CDC and the WHO lists, as well as the syndromes that we know are driving the greatest morbidity and mortality globally.
Our mission is to change the way infectious disease is diagnosed and treated. So in order to do that, we're building a new technology that makes use of whole genome sequencing and machine learning to have a faster way to diagnose infectious disease. And we do that whole process in 8 hours rather than in the 3 to 5 days that it might take in today's world to get those results.
CARB-X currently supports VE707, which is a drug that we have in preclinical development for prevention of antimicrobial resistant infections for E. coli and Klebsiella pneumoniae.
Vedanta has a novel approach using live biotherapeutics, and the idea is that these live biotherapeutics will decolonize the gut of harmful bacteria that could break through and cause infections.
CARB-X works in lockstep with us and provides very rapid access to staff that they have on board already who have expertise that's directly relevant to our program, and their network of advisors who are ready and willing to come in to help product developers like us.
We build teams around them to augment their expertise so that then ultimately we bring the products that they're developing closer to patients.
There are two critical areas that CARB-X collaborated with us to advance VE707 and really accelerate the development. The first was in the discovery and evaluation of the spectrum of activity of VE707. One other area that they really accelerated the program in is connecting us with clinical experts to design our first-in-human study. Without CARB-X support, it would have been much more challenging to get VE707 ready for evaluation in a first-in-human study.
In the Day Zero case, we were looking for technologies largely focused on bloodstream infections, so diagnosing bloodstream infections. And Day Zero has a very unique approach, and so it had a unique technology and it fit the syndrome for which we wanted a diagnostic.
We have been very fortunate to be able to work with CARB-X. They've really become, just really development partners for us, working together to figure out how to push the technology forward, and over time we've iterated with them and pushed together with them to bring the company to a more mature technological stage and ready for kind of the next stage of things like FDA submission and thinking about going out to market.